Session will be about polygenic risk scores. The first speaker is Barbara Domingues Bitarello, who is a computational biologist and geneticist from the University of Pennsylvania. And she is interested in all things related to human evolution. She has worked with immunogenetics and balancing selection. And lately her focus is on polygenic risk scores, particularly on admixed populations. And this is also what she will talk about today, I think, if I'm right. So Barbara, the floor is yours.
Okay, let me share. Okay, if you don't see a full screen with my slides, please let me know. I think it's good. So good afternoon, everyone, or in my case, good morning, late morning from Philly. I'm a postdoc at UPenn, as was just said, and I've been very focused lately on the issue of transferability of polygenic risk scores across ancestry. In particular, not only across ancestries, but for admixed individuals and populations in general. So that's really what I'm talking about today, mostly focusing on recent work we published in October now. Sorry, okay, yes. Okay, so I'm from Brazil, so this is a painting that I like. And I like to start by saying that we as a species, we have diversity in behaviors, in traits like appearances and some disease predisposition and that run in families and things like that. Some of the diversity we observe in our species is rooted in genetics, and that proportion that is rooted in genetics is the subject of obsession of population geneticists. We want to learn about this relationship between genetics and phenotype. So ever since we as a community sequenced the first human genome, which was not that long ago, we started doing these so-called genome-wide association studies. And so for the past 20 years, we've done a lot of these studies, hundreds of studies like this, where basically you have hopefully a large cohort of individuals. If you're looking at a binary trait like absence of a disease or presence of a disease, you would have a control and a case group. And if it's a quantitative trait like height, you would just have a big sample of individuals. And hopefully you get their DNA and you measure the phenotype of interest. So let's go with height because I like height. And but you also hopefully measure some covariates as well that you know influence that phenotype and that are not necessarily genetic themselves. And then for each of those variants, because you have this large sample size, you can test for association of each variant in the genome, an association between the phenotype and the variant. And then this is just a schematic, and it would be a very large effect, but it's showing that if you have a 0 genotype, you have a certain phenotype. And if you have the 1 or 2, you monotonically decrease this phenotype. So each variant will have a beta or an odds ratio if it's a binary trait. And then from that, you typically plot these results as like a Manhattan plot where you have each variant has a p-value, and based on a lot of complicated statistical procedures, you decide what's significant and what's not. And hopefully at the end, you will have some candidate SNPs or that are either causal or are tagging causal variants that are associated with that trait. One picture that has emerged from 20 years of GWAS is that most variants that we're able to detect, they lie right here. So they are intermediate in frequency and they have moderate effects. Now, what we really want is like big effect size. We want these guys, right? Highly penetrant mutations. But in general, these are rare. So that's why we don't often get them. They often run in families and they have huge effect sizes, but they are not very common. There are some exceptions. For instance, the APOE4 variant for Alzheimer, it falls somewhere around here. I hope you can see my cursor. I think so. It falls somewhere in the gray area here. So There are exceptions, but these, we get very excited about them, but they are rare. So this was somewhat disappointing early on. And as we've increased GWAS sample size, we do get more variance. And we've been able to explain more and more proportion of variance in phenotype, but it still falls very short of the initial goal or expectation that researchers had. But instead, we found out many important things. So one of them being that most traits are polygenic. And that is why we detect so many variants and they have moderate effects. So the first time this was done was in a schizophrenia GWAS from the early 2000s. Researchers found very little in terms of like big effect variance, but they decided then to combine all the significant hits in the GWAS, so that didn't quite make the threshold. They combined it into a polygenic score. And they were then asked, if we combine all these variants, can we explain some variance in the phenotype? And it turns out they could. And this implementation is rather simple. Its basic format is it's just a weighted average of the betas. So those effect sizes I mentioned, you use them to weigh the genotypes. So each individual has a PRS for a trait. And that hopefully can give you a sense of the individual's risk or predisposition for a certain trait. And this is pretty easy to implement in terms of math. It's fast. It has minimal requirements in its simpler forms. Like you basically need summary statistics from a GWAS. You don't even need to run the GWAS yourself. And as a result, there's a lot of hype around this. So there's like, as we were discussing just now before this session, there's like a lot of papers coming out with polygenic scores, and not all of them are very careful in their conclusions, and but some results are very promising. For instance, this paper from Kira et al. From 2 years ago is already a classic in that they showed that when you calculate polygenic risk scores for cardiovascular disease using variants across the genome, so not only the significant variants from the GWAS, but just using all variants, you can then bin individuals according to their percentile of PRS. And the individuals in the higher bins here, so the individuals with the highest PRS, they have a prevalence of more than 10% of cardiovascular disease. And that's comparable. This is in magnitude, this is comparable to individuals with large monogenic mutations. So mutations with large effect on this phenotype. Which typically run in families. So some people think this is actually disappointing because it's not better. But actually, this is very promising because these variants that have large effect, like I mentioned the Alzheimer one, or the BRCA variants for breast cancer, they say a lot about your increased risk. But so having them says something, but not having them doesn't say anything about your risk, right? Whereas, and they are somewhat rare apart from certain populations where they're more common, but polygenic risk scores, everyone has one. So as long as you have a decently sized GWAS and you believe the effect sizes, you can calculate a polygenic risk score for any individual. And even if you can't quite predict phenotype on an individual level, if you're able to bin people into risk groups, as they did in this paper, this is actually very promising. And another promising venue is to use these bins to choose people for GWAS, which is also a very interesting perspective. So I do believe that there's a lot of promise in using this kind of approach. And here are some more examples. So for height, which is a highly heritable trait that we understand well, we can predict up to 25% of the variation in height using these polygenic risk scores. And we found more than 3,000 independently associated variants with height in the human genome. And even for neuropsychiatric conditions that normally there's a sense that they're more complex, the effects of the environment are really hard to pinpoint, and indeed they are, but even there you can explain up to 7% these days with the GWASs we have. And these numbers keep increasing as we increase GWAS size, and there's a lot of theoretical work showing how far we could get with those approaches. And finally, for binary traits like breast cancer and cardiovascular disease, we also have good results. So these are like the best examples, arguably, and all of this, everything I just presented, was exclusively done in, I should say not Europeans, but European ancestry cohorts. So, and this is the goal of my work, and I think of my ongoing work for the foreseeable future. Like what about ancestry? So we know that human populations we know a lot about our demographic history. We've accumulated some differences on the continental level because of shared drift, really, because of being isolated from each other and drifting in different ways because we were isolated. So there are systematic differences in frequency spectrum and LD patterns across populations. So of course they will affect things like your ability to tag a causal variant with a GWAS and your performance of a polygenic risk score. So I want to send a clear message here about the lack of representation of human diversity in GWAS. So GWAS are invaluable resources, but in this figure that is now already a classic from last year, Sirugo and Tishkov, they show that about more than 78% of participants in all the GWAS that we've ever done are of European ancestry, and only about 2% are of African ancestry. Most of the others, the other 20%, are East Asians, and that is rather recent. 10 years ago, this number was 96% European ancestry. So there has been some improvement, but it falls very short of what we would actually need. So as a comparison, which is a loose comparison because this is not ancestry, but this is just population. This is from World Population Review. So I plotted this pie chart, and it's showing what's the percentage of people that live in each continent. So Asia harbors 60% of the human population. Europe harbors 9.6%. And so even if we assume that everyone in Europe has European ancestry, plus some percentage of North Americans and other people in the world, it would still fall short of 15% of the world's population harboring European ancestry. And then you look at this 78%. And this is, I'm just talking about continental populations, not to mention that, that a big proportion of the world population these days is actually admixed. So Latin American populations are largely admixed between European and African and Native American. So, and that kind of ancestry is even less represented than the so-called discrete groups. Okay, so this was the focus of my research. At the time, no one had really looked at accuracy of polygenic risk scores in other ancestry. There was very little. In the meantime, two important studies came out. This one by Alicia Martin and collaborators, where they pooled 17 traits, 17 quantitative and binary traits for humans, and they measured the prediction accuracy from polygenic risk scores. So shown in black here is the European as like a normalizing factor. And then you have these other populations. I'm not really sure what American is, but I'm assuming it's North American. So not the best one to look at, but South Asian, East Asian, and African are really like populations from those regions. And you see this monotonic decline of accuracy, and with Africans being about 25% of the prediction accuracy in Europeans. And this is an average, so it does vary a bit depending on the trait and the architecture of the trait, but this is roughly how it is. And then in this very interesting paper from this year by Wong and Vischer, they show this is a theoretical and simulation-based work. So they simulated a number of architectures, so represented by M here, like number of causal variants and heritability on the rows here. But the important message here is that they see on the x-axis, you have FST between Europe and other populations. So as the FST increases, you have this decline in accuracy, and it's predictable if you assume that heritability and causal variance and everything is shared across populations. But just because of the differences in FST, so site frequency spectrum, basically, we would see this decline. So this is very interesting because it gives a quantitative sense of that we should expect this to happen. It doesn't solve the problem at all, but it's a documentation that it was bound to happen because of the way we have done these studies in focusing only on European ancestry. But empirically, there hasn't been much in terms of like what is actually going on that causes this lack of prediction. So, and understanding that could maybe let us fix it. So is it a problem of SNP genotyping, you know, so-called like this European ascertainment bias, which has been much better in recent implementations of SNP chips? Like, what is happening, and can we leverage things differently in these scores to account for it? So this is not an easy task because many factors may influence PRS. So don't be misled by the simplicity of the math of the polygenic score. There are a lot of assumptions that go into it. So for instance, even in the same ancestry, there's like paper from Molly Zaworski this year showing this. Even within the same ancestry, if the cohorts differ a bit, you already get very different prediction accuracies. So then when you go to different ancestries, you could have local selection in one population, but not the other. The LD and site frequency spectrum differ, and that's a fact. So that will have influence, that will affect the additive genetic variance. Phenotypic variance might be different. You might have local gene interactions, gene-gene and gene environments that are different across populations. And that's all even if you assume that the causal variants are the same. Not to mention that maybe there are a few that exist in one population but not the other, or are at very low frequency in Europeans, for instance. So the task is complex, and these factors are not mutually exclusive, but I set out to try to uncover what was going on. So in terms of what factors affect the accuracy, my approach was to see how well we could predict a phenotype in a gradient of ancestry. So instead of looking at ancestry as a discrete thing, like European, African, more like a gradient going from European to African, which was the object of my study, and then try to pinpoint where the inconsistencies happened, and then try to approach, try to account for ancestry once this was pinpointed. But it was very complicated, actually. So let's look at height, which is what I looked at in this paper. Why height? So height is very heritable based on twin studies, 80% heritable, and based on SNP heritability in European ancestry, typically about 50% in most genotype arrays. So what we did here is we took the summary statistics for height for over, it's like 360,000 individuals from the UK Biobank. They all have European ancestry. So this is the largest GWAS for height so far. And so we have the effect sizes, and we know that they're all coming from European ancestry. And then we have our test cohorts, which were about 20,000 individuals from different data sets, most of which were African-American cohorts, so meaning they had European and African ancestry admixed in each individual. And some of them, for control purposes, were European as well, but independent from the discovery cohort. So the European cohort we had was the HRS, Health and Retirement Study, European individuals, European ancestry individuals. Everyone else was mostly admixed. So what we had, and as I mentioned, we looked at ancestry as a continuous variable. So for each of these 29,000 individuals, including the ones we're calling European here, they had between 0 and 100% European ancestry in their genome. And the way we assessed that was we did local ancestry inference for each variant in the genome of each individual. So when we say an individual had 25% European ancestry, we're averaging the local ancestry inference. Okay, so then we looked at how well we could predict height in these individuals. And what we see, and is the main message from this paper, is that there was a remarkably linear relationship between how much European ancestry you harbor in your genome, regardless of where, just the average proportion in the genome in the x-axis, and the amount of variation you can explain by the PRS in the y-axis. And the different colors are the different cohorts, which I'm not putting their full names here for simplicity, but they're mostly from the UK and the United States. So the black square here is the European ancestry cohort from the United States. That is like our baseline. All the other ones are admixed, and there's different number of dots or for points for each data set because we binned individuals according to European ancestry. So instead of just looking at the entire data set as one number, we binned individuals. So each bin had about 2,000 individuals, and we have the confidence intervals. It's remarkably linear, and we can even predict that at 0% European ancestry, you would only predict about 1% of variation in height compared to like 15% here, 12%. And for each 10% increment in European ancestry proportion, you get about 1% increment in variance explained. So this is very interesting, and it was somewhat surprising that we saw this like 74 to 80% reduction, and you know, the theoretical work I've mentioned to some extent predicts that we would see such a reduction, although they document less of a loss than this. So you know, reality is always more complicated. So then we tried to uncover what was really going on. And surprisingly, as I just showed in that figure, that relationship was very linear, close to what we have in K equals 1 here. So this is just this equation here, Y being the phenotype, and I mean being the partial R squared, and 0.15 is just the slope. So we actually, when we look at only European chunks of the genome, so this plot is different from the previous one. The previous one, we were looking at all chunks. Here, we're looking at only the European chunks and then doing the same analysis. So let's say this individual here in red has about 40% European ancestry proportion, and we're only looking at the European chunks here. So you actually fall short of this K equals 1. So this is very interesting because it's suggesting that not all predictive power is coming from the European chunks. Yes, there's an overall linear relationship with ancestry, but it's more complicated than just picking the chunks and then calculating the risk scores based on those chunks, which probably explains why when we tried to leverage local ancestry, we didn't get very amazing results, which I will show. And in K equals 2, which is this line at the bottom, would be what you would expect if predictive power doesn't come just from European chunks, but it's rather spread uniformly across the chunks. We see something in between, and there's some error associated with it. And also, I should point out that Marnetto collaborators, they state in their paper from this year that they can definitely recover this linear trend just by using the European chunks. So perhaps it's still a bit unclear what is going on here. So we use different approaches. But I just wanted to mention that. Okay, so next we have the issue of site frequency spectrum. We know there are differences. We know genotyping arrays also have their biases. So we asked whether the additive genetic variance was different. So let's focus on this figure in just the top part that says HRS. So what we have there is a ratio of genotype additive genetic variance. So the numerator is for the African ancestry cohorts and the denominator for the European ones. When I say African, I mean the admixed ones. And we see that at most, so if you get a value close to 1, it means there are no systematic differences in site frequency spectrum in the variance that you're using for the polygenic score. And in fact, we see that it's about 90% here. So you do, and in this other comparison here, the biobank, it's up to 20%. So at most, you would explain 20% of loss of accuracy based on this. So there's definitely that effect, and this is very dependent on each data set. We didn't have, you don't always have a comparison of like African and European for the same cohort. This will depend on SNP chips and other things. So older SNP arrays have more bias for common variation in Europeans than the more current ones. So this will definitely depend on each study. Next, we thought, okay, maybe there is actual phenotypic variance that is different. So we know from population genetics that just genome-wide genetic variance is lower in Europeans compared to Africans. So it's about 3/4. So if that is also true for the PRS variance, then perhaps inheritability and environmental effects are the same, then you would expect less phenotypic variance in the 100% European bins. So in this plot, on the x-axis, we have European ancestry. On the y-axis, the standardized residual height. and it's only very, very slightly increasing. It's not even significant. And what we plot here is that the dotted gray lines, which you can't see because the blue one is overlapping it, is just the standard constant variance. And then we have this more elaborate model shown in orange, where we allow for phenotypic variance to decrease as a function of European ancestry, according to the 76%. And finally, we just fit this model, also making the variable free and allowing for it to fit the model itself. And it fits the model at very close to standard deviation of 1. So basically, we don't find any evidence that phenotypic variance is lower for European. That is the main result here. So this does not seem to be driving the difference. Finally, you have this issue of linkage disequilibrium. I'll go fast through this because I'm running out of time, but you know that when you get a variant in a GWAS, it's either the causal variant or it's tagging a causal variant, as shown here by the orange and yellow, because they're in tight linkage disequilibrium. If in another ancestry, those two variants are not in tight linkage disequilibrium, then you could fail to get to tag the variation appropriately. And this could lead to a systematic decrease in accuracy. So first we saw that if we use imputed data instead of genotype data, we don't see any difference in this pattern. So assuming that imputed data, you have a higher density of SNPs and that's better, we actually don't see a difference, which means that the genotyping arrays are capturing the relevant variation. So this is not the issue. Next, we tested whether variants falling in low regions of recombination were, if we produce PRSs just for each bin of recombination in the genome, whether they were more transferable or not. And we expected that if loss of accuracy is independent of LD, we would see the same pattern happening in both ancestries, regardless of the direction, because it could go up or down depending on genes and other things, but you would see the same pattern. Whereas if it's dependent on LD differences, you would see that at low recombinations, you would have similar performance. And as you increase recombination, you would have different performance. And we do see that. So in this plot, you know, the colored bars are the non-European cohorts. It's somewhat going down as you increase recombination, whereas the black ones are going up. So if we focus in low recombination regions, things are more similar in terms of like relative contribution to the PRS. But the absolute difference here is still about 70%. So the main message here is LD does play a role, but it's definitely not enough to explain that linear trend I showed, at least not in our studies. So, and finally, we also try to estimate effect sizes that were African specific by taking 9,000 African ancestry individuals from the UK Biobank and running a GWAS. This is very underpowered. We did it nevertheless, and here are the effect sizes and their correlations with European effect sizes. And we saw that the difference, so the effect sizes that are more different across these ancestries, they tend to also have more systematic differences in allele frequency across populations. And this seems promising, although it's a small correlation, but it's significant. So this suggests that there are differences in effect sizes across populations. It's not just a matter of tagging. And finally, assuming that there are these differences in marginal effect sizes, it would be nice if we could weigh these different PRSs coming from different ancestries in the admixed individuals. Now, this is not new. It was done by Marcus Luna 2 years ago. We re-implemented it, but we also did 2 other variations. So these are all linear combinations of a PRS coming from European ancestry and African ancestry, but in Marcus Luna approach, you weigh everything by alpha and 1 minus alpha, like the entire cohort. Whereas in the 2 that we devised, you use each individual's proportion of European ancestry also. And in the more complicated one here, the 3rd one, you use the local ancestry inference. So for each variant, you decide whether you're using the European or the African beta. And we thought we would see a big improvement using that.
1 more minute.
Yeah, this is the last slide. Yeah, so we thought we would see a great improvement, but it was only like about 1.2 at max, not even 1.2-fold improvement. We really believe that this is largely due to our very small GWAS for African ancestry, 9,000 individuals. And in fact, I've been doing a larger GWAS now for that, and we do get something around 1.5 improvement. So it's still much lower than what you see for European ancestry, but part of it was our small GWAS. So what I want you to take home is that at present, PRS have no utility for ancestries other than European. So when you do your 23andMe or whatever, take it with a grain of salt. And many factors are involved here. It's not easy to fix it, and we really need to diversify genetic studies. From my end, I'm still working on this, optimizing ancestry-sensitive approaches, and working with researchers from Latin America, trying to focus on those populations now. And as I mentioned earlier, with colleagues, we really should try to make LD reference panels available for other ancestries, even if we don't provide the genotype, but just the LD matrices, because this would allow for more people to use these elegant PRS methods and explore other populations. And I really do believe, based on our findings, that there are some population-specific gene interactions at play that would also be interesting to analyze. And everything I showed here should, to some extent, happen for other traits, but it could be more or less complicated depending on architecture. So I'm sorry I went a bit over. I would like to acknowledge Ian, my mentor, and the lab, and everyone that contributed data to these studies that are all publicly available. Thank you, and if you have questions, since we don't have a lot of time, please write to me or talk to me in the session after. Thank you.
Barbara, thanks a lot for the intriguing talk. So we know from rare variants that the African continent is actually particularly interesting because of its high heterogeneity. So could you comment on your thoughts about the value of this high heterogeneity also for PRS? Do you think there might actually be a gain of accuracy if we sample more Africans?
Yes, I do, because there's actually independent work from my own showing that if you have a more diverse GWAS to begin with, you do get better prediction, not for the European ancestry, but for all the other ancestries, you see an improvement. And I myself have been doing that now, and you definitely see an improvement. So even small numbers of other ancestries does already improve this, because there's just variation you're not detecting at all in a study like this. And it might not be relevant for the population that you're looking at, in this case, European, but it might be for all the other ones, because these variants segregate at different frequencies. So definitely, and there's a lot of proof out there that that is the case.
Okay, thanks a lot, Barbara, again, and hopefully we can continue the discussion in the Q&A session. And now we